Hello friends, we discussed about microbial history, diversity and taxonomy in previous video. We discussed about historical developments and scope of microbiology, concept of spontaneous generation, biogenesis and Francisco Reddy's fly experiment and Louis Joubert. We also discussed about John Needham. Now let's move further. Let's learn about Abbe Lazzaro Spallanzani. Spallanzani found that sealed flasks containing infusions heated for 1 hour showed no cloudiness after a reasonable period. Spallanzani responded by breaking the seal on his heated closed flask, allowing exposure to air. Within a short period, the contents of this flask became turbid, showing that the long-heated organic matter was still capable of supporting life. He found out that organic matter, after heating long period of time, still supported life. Then this study was further continued by Theodor Schwann, set up 2 separate flasks containing infusion. In one flask, air was passed through a red-hot tube before entering the flasks containing the nutrient material. The flask remained free of any growth as heating destroyed all life forms. In another flask that received unheated air, soon growth developed. When he performed experiment, one flask was sterile but other was not sterile, so unheated air results to growth. Then next was Franz Schulze. In his experiments, in one jar he passed that the air to pass through strong acid like a sulfuric acid, sodium hydroxide before entering into nutrient or meat. No growth developed in this flask, while in other that received untreated air, soon growth developed. Growth was obtained because of untreated air. Conclusion. So what he concluded, above experiments concluded that the treatments of the air with heat or chemical destroys all possible life generating power, but untreated air results growth. Now let's see what Henry George Frederick Schroeder and Theodor von Dutch introduced. Use of cotton plugs for bacterial culture flasks and tubes. Using Schulze and Schwann system, these scientists allowed air to enter untreated in any way except by being filtered through cotton wool that had been previously heated in an oven. Flasks that received filtered air showed no growth, while flask without cotton plugs receiving unfiltered air contained microorganisms. Again the same thing, they tried to protect from cotton plugs, but growth was appeared as unfiltered air contained microorganisms. Now what Pasteur's experiment showed us, let's learn. Pasteur passed large volumes of air through a tube with a plug of gun cotton as a filter. A piece of gun cotton was then dissolved in an alcohol ether mixture, and the remaining sediment was examined under the microscope. Pasteur found small round and oval bodies that resembled the spores, reproductive structures of plants, to show that the gun cotton not only stopped the passage of microorganisms but contained these life forms of life as well. Pasteur simply added a little of used filter to sterile meat infusions. Soon microbial growth appeared. Thus Pasteur confirmed the ways in which microbes gained access to nutrients and how they could be prevented from doing so. Nutrient liquid media were placed in the flask, and both the container and liquid contents were sterilized by boiling. No plugs of any type were used to prevent the passage of microorganisms in these systems. Microorganisms present in the air could not reach the main part of flasks due to the large length and the end of the flask's Schwann neck. When a top of system was broken off, or if a flask were titled so that the sterile liquid nutrient ran into the exposed part of the necks and then returned, microbes soon appeared in the fluids. As we can see here, tube is seen, Schwann neck, microbes are being destroyed from the flame you can observe, and in the next diagram we can see here the growth stops from getting affected in the flask, and here we can see